but I did want to take a moment here to introduce Matilde Kam. We're so grateful that she's a part of RN Pharma, and without further ado, Matilde, if you'd like to begin your presentation, we're excited to hear what you have to say.
Okay, Daniela, can you hear me?
Can you
see my slides? Okay, cool.
See your slides.
Good. Okay, thank you.
Thanks.
Good morning, everybody. I'd like to thank the organizers for inviting me to give this talk. My name is Matilde Kam, as Daniela said. I am the Associate Director for Analytics and Informatics of the Office of Biostatistics at the Center for Drug Evaluation and Research at the FDA. The title of my talk is Challenges We Face in an Evolving Regulatory Landscape, How Statisticians and Data Scientists Can Contribute. So here's my disclaimer. The views and opinions presented here are my own and should not be considered to represent advice or guidance from the FDA. Here's the outline of my talk. First, I'd like to provide an overview of the Office of Biostatistics and my group, the Analytics and Informatics staff. Then I'll go over the various FDA initiatives that we have and the challenges we've encountered. And lastly, I'll provide some summary statements. So the United States Food and Drug Administration is composed of 9 centers. I work for the biggest center, which is the Center for Drug Evaluation and Research, or CDER. And these are the other centers, although I didn't put all the 9 centers here in the diagram. So we have CBER, CDRH, CTP, CIFSAN, and CVMs. These are some of the centers at the FDA. So the Office of Biostatistics is part of the Office of Translational Science, or OTS, which is one of the CDER super offices. So the other offices under OTS are the Office of Computational Sciences, the Office of Clinical Pharmacology, which houses the clinical pharmacology reviewers, the Office of Study Integrity and Surveillance, or OSIS, which is the office that conducts and directs the inspections of bioavailability, bioequivalence, and non-clinical GLP studies that are submitted to the FDA. So the Office of New Drugs is another of CDER super offices, and they house the medical officers and clinical reviewers. So here's the mission of the Office of Biostatistics, which I will not read, but inherent in this statement is OB's responsibility to promote and provide strategic, quantitative thinking to medical product regulations, not just to keep an unsafe or ineffective drugs from the market, but also to speed proven therapies to market through the use of state-of-the-art statistical methods. So the Office of Biostatistics has about 240 statisticians spread out across the different division of biometrics. So DB1 to DB5 and DB9 work with specific therapeutic areas. DB6 deals with non-clinical studies. DB7 deals with special safety studies, and DB8 deals with generics. Now, traditionally, majority of our statistical reviewers are PhD statisticians. So unlike in industry, our statistical reviewers take care of everything. They do their own data setup, data manipulation, programming during the reviews. Now, with CDER's modernization effort that encourages integrated, collaborative, issue-based reviews, OB statistical reviewers are now engaged more in safety and benefit risk evaluation in addition to efficacy. So to be able to contribute effectively to the modernization effort, we need to bring on more people. So with the added safety and benefit risk responsibilities, the Office of Biostatistics decided to create a new group comprised of master level statisticians, or what we call internally as statistical analysts. So we found that PhD reviewers were spending substantial time on tasks that could be done by the statistical analysts. So that new group is called the Analytics and Informatics staff. So this is my group, and we report directly to the immediate office of the Office of Biostatistics. So the OB, they call us AIS. So the OB-AIS group was formed to be in the forefront of addressing OB's emerging needs in analytics and informatics. So we provide leadership and consistent support to the 9 divisions of biometrics in terms of data standards, data integrity and data quality, data science, data tools, scientific computing, statistical programming, and project management. So statistical analysts provide support to statistical reviews in NDA-BLA, statistical reviewers in NDA-BLA reviews, and special projects. So these changes are intended to free up the PhD statistical reviewers' time, so they have more time to think about the issues as well as engage in safety and benefit risk assessment. So for this talk, I'll go over some of the challenges in the evolving regulatory landscape. So significant advances in science and the availability of new data sources coupled with modern technological innovations have increased business and competitive opportunities in the pharmaceutical industry. So this has occurred in the search for better and cheaper medicines that reach patients with unmet medical needs as quickly as possible. This provides an excellent opportunity for statisticians and data scientists to take the lead and help business and regulatory leaders make good decisions regarding drug development and market authorization as quickly and efficiently as possible in the presence of uncertainty. So remember, we statisticians and data scientists are at the heart of evidence collection, synthesis, and communication. So for the rest of this presentation, I will discuss FDA's innovative approaches and initiatives to meet the challenges of evolving science, technological advancement, and new data sources. So I'll go over the regulatory and statutory basis of the various initiatives, and then I will highlight areas wherein statisticians and data scientists can contribute and play a key significant role in medical product development and evaluation. So one of the statutory bases is the 21st Century Cures Act. This was signed into law in December 2016 and is designed to help accelerate medical product development and bring new innovations and advances to patients who need them faster and more efficiently. So this law builds on FDA's ongoing work to incorporate the perspective of patients in FDA's decision-making process and enhance our ability to modernize clinical trial designs, including the use of real-world evidence, clinical outcomes assessments, which will speed development and review of the novel medical products. Another thing is on August 2017, the FDA Reauthorization Act was signed into law. And this new law includes the reauthorization of the Prescription Drug User Fee Act, or otherwise known as PDUFA. So now it's its 6th iteration. So this is called PDUFA 6. So that's how we usually say it. So this is valid from 2018 to 2022. So this provides FDA with the necessary resources to maintain a predictable and efficient review process for human drug and biologic products. So some of the PDUFA 6 goals that I'd like to highlight are, enhancing the use of real-world evidence for use in regulatory decision-making, enhancing the incorporation of patient voice in drug development and decision-making, enhancing the capacity to review complex innovative designs, and advancing model-informed drug development. So let's look more closely into CDER's new drug regulatory modernization program. So in 2017, FDA CDER embarked on an initiative to modernize the new drugs regulatory program. This effort will be long-term. It will be a long-term process of continuous improvement involving multiple initiatives. So one of the initiatives, which I already mentioned earlier, is the integrated review of drug marketing applications. And as part of this initiative, CDER is implementing a streamlined, integrated, interdisciplinary review process that for the assessment of marketing applications. So the goal here is to strengthen the clarity of the benefit risk assessment, as well as to improve communication concerning approval decisions. So in tandem with this integrated review process, during the past year, several offices in CDER, including the Office of New Drugs and the Office of Biostatistics, were reorganized for greater efficiency. So within the Office of Biostatistics, we are measuring and standardizing workflow, roles and responsibilities, and identifying opportunities to improve our operational efficiency. So the goal of the standardization is to allow our statistical reviewers to focus on statistics and our statistical analysts to focus on developing and maintaining standards. Now, this includes standard statistical programs and standard processes that can be applied across the review divisions of the Office of Biostatistics. So we're trying to create consistency across the 9 divisions that we have. So now let's look more closely into the FDA's real-world data and real-world evidence initiative. So as part of the PDUFA 6 and the Cures Act, FDA has created a framework in 2018 for evaluating the potential use of real-world evidence, RWE, to help support the approval of a new indication for a drug already approved, or to help satisfy post-approval study requirements. So last year, FDA also issued a guidance on submitting documents using real-world data and real-world evidence to FDA for drugs and biologics. So FDA, here's definitions on what we mean by real-world data and real-world evidence. So real-world data are data relating to patient health status and the delivery or the delivery of healthcare routinely collected from various sources. Real-world evidence is the clinical evidence regarding about the usage and potential benefits or risk of medical products derived from analysis of real-world data. So for real-world data, some of the sources that we're looking into are the EHRs, medical claims and billing data, or data from registries or patient-generated data in home use settings, and data from any other source that can inform the health status, such as mobile devices. So the FDA real-world data, real-world evidence emphasizes FDA's 3-part approach to real-world evidence. So the first part is evaluating whether the real-world data are fit for use. Second is the trial or study design used to generate the real-world evidence provide adequate scientific evidence to answer or help answer the regulatory questions. And third, does the study conduct meet FDA regulatory requirements? So these are the 3-part approach. And outside of that is looking into data standards and implementation. So what are the goals for the use of real-world data, real-world evidence? So here is an example of a schematic for a traditional clinical trial with defined visit schedule. So as we all know, a randomized controlled clinical trial is the best way to minimize bias in ascertaining treatment effects. So here we typically use selected group of patients and have special infrastructures to conduct the study and collect data. However, in doing this traditional RCTs, the periodic encounters may not provide a complete understanding of a subject's experience or overall health during the investigation. So, for example, if we're in a diabetes study, marked fluctuations in the glucose levels of the subjects might not be taken or will be missed in a traditional trial. So, because we only see them at specific visits. So on the other hand, the use of real-world data would reflect the diversity of patients and actual healthcare practices. So it improves efficiency, but making use of existing data and infrastructure. So however, even if we're allowing the use of real-world data for real-world evidence, we have to keep in mind that studies that use real-world data should still maintain evidentiary standards as required by the FDA. So the standard for drug approval remains the same. So here's an example on how our real-world data is typically collected. As you can see, there's no defined visit schedule. So information regarding a patient can be found in different databases, such as a claims database or an electronic health records, or we might not even be able to capture it if it happens outside of these encounters. So this is a graphic that provides the various considerations considerations we have across the real-world evidence spectrum. So it ranges from a traditional clinical trial that uses some real-world data elements, for example, in the enrollment criteria or trial feasibility, all the way to a heavy reliance on real-world data for observational studies. So, it could be non-randomized or non-interventional. So this is a good graphic that shows you the different spectrum. So in this slide, you will see the various considerations that we should take into account if we're thinking of using real-world evidence. So statisticians and data scientists can have a significant impact in addressing, the data considerations and the different methods consideration. So for example, some example of it is how do you check the reliability of the data or the validity? So how do you impute the missing data? So these are some of the questions you should look into if you want to use real-world evidence in your submissions. Now, here are some of the challenges with the use of real-world data or real-world evidence in clinical trials. So they include, for example, how do you define an estimate, or how do you estimate the treatment effect which is more complicated in a real-world data setting? So how can we combine different data sources? How do we determine data quality? So what methods can we use to address the duplication of patient information in different data sources? So if the information of a single patient can be found in different data sources and we combine those data sources, we have to make sure that we don't double count the patient. We also, how can we use machine learning to produce regulatory grade evidence from real world data? How can we utilize, natural language processing or NLP to get more insights from unstructured data sources? So what can when can we use a historical control, or concurrent controls, or even synthetic controls? So what is the best way to control for confounding or adjusting for bias? So to come up with solutions for this different issues requires collaboration among the various stakeholders and the development of software tools to implement the newly developed analytical methods. Now, the use of open source solutions, such as those developed using R, will help facilitate these collaborations. So now let's look into the Digital Health Technology Initiative. So again, the 21st Century Cures Act includes a provision related to the digital health technologies. So when we say DHT, we refer to a broad category of technology relating to health applications. So the framework of the FDA's real-world evidence, which I discussed previously, recognizes a variety of clinical trial designs, ranging from the traditional clinical trial to a fully decentralized clinical trial. Now, DHTs may play an important role in this novel settings. So, as I've said, we have different centers at the FDA. So the FDA Center for Devices and Radiological Health, or what we call CDRH, is the main center responsible for regulating medical devices. So any clinical investigation in which the DHTs are being evaluated as a medical device falls under their purview. So CDRH has issued a Digital Health Innovation Action Plan in 2017, as well as last month, FDA announced the formation of a CDRH Digital Health Center for Excellence. And this center will align and coordinate digital health across the FDA. However, the focus of what I'm going to talk about next would be on DHTs measuring clinical endpoints or physiological data in clinical studies, and not on having DHTs as medical devices. So why use DHTs in clinical trials? Well, they can help minimize barriers in obtaining patient experience data during clinical investigations. So here, the ability to capture continuous, frequent, and real-time remote data may improve the understanding of the drug effects, as well as the relationship of these effects to the activities of daily living. So that's an advantage of using a DHT in a study. It could also streamline study and data monitoring procedures. So DHTs may be used to potentially reduce the missing data by sending automated reminders to the subjects to take their medication, complete an e-diary, or attend a follow-up appointment. It could also help maximize recruitment efforts among hard-to-reach, geographically diverse or dispersed or rare patient population. So those are some of the advantages of using DHTs in clinical trials. Now, here are some examples of endpoints measured or acquired using DHTs. They can serve either as a clinical outcome assessment or a biomarker. So this is an example of a DHT that provides a large amount of continuously collected data from a study subject over time. So this is just an example of one type of wearable 8-minute of data. So this is just 8 minutes of data. So as you can see, there's a lot of data that we would be getting from DHTs, and that's where we as statisticians and data scientists can have a lot of impact on how do we process this data, how do we summarize this data, how do we analyze this data. So here's another example of this is more of an example of a daily activity count for a given subject. So each of the curves represent the activity count at the minute for a given day. The red curve represents the mean of the daily curves. And, the questions we might be asked here is, so how do we summarize this data? Do we look at the entire curve, or do we focus on a specific part? So which features of the curve do we focus on? Okay. So here, so this graphic provides some of the key considerations in the design, implementation, and analysis considerations for the use of DHTs. So I want to point out that study endpoints, whether the traditional one or derived using a DHT, as long as they're going to be used to support regulatory decision-making and labeling claims, they must be based on well-defined and reliable assessments. So, as I said, here are some key considerations that we should think about. We need to think about the verification and validation of the DHT to ensure that the accuracy and precision of a DHT in performing its intended function. So how do we collect the data? What variable are we measuring? Are the endpoints we are planning to use well-defined, reliable, and clinically meaningful? We should pre-specify the statistical methods we are going to use to process and analyze the DHT data. Also, it's more likely to have more missing data if we use DHT to measure our endpoints. So now the question is, how are we going to handle missing data? How are we going to impute them? So those are some of the questions that we have to take into consideration. And here are some more specific challenges in using DHTs in clinical trials that we can help with. So how do we validate or verify the DHT? One thing that we need to have if a DHT data is submitted to for regulatory evaluation is we have to establish the provenance of the DHT data. We have to know where it came from where it came from up to after it has been processed. So that's one thing that we look into. Another thing is we need to have a clear understanding of the method used to process the DHT data, such as the machine learning algorithms. We have to have, complete disclosure of what you guys use to process the data. Another is we need to develop data standards for DHT. Currently, there's no standard format for analysis and summarization of continuous data for DHTs. In addition, summaries, for example, activity count, steps, or calories, although they have the same name on different DHTs, they might have different meanings or different ways of deriving those summaries. Also, companies do not want to disclose proprietary algorithms. So it's hard to figure out what exactly their device is providing. Also, it's difficult to translate or generalize results we've obtained from a given DHT. So those are some of the questions we should look at. So here, so let's now look into the Complex Innovative Design Initiative. Again, based on the PDUFA 6 commitment and 21st Century Cures Act, we have a commitment related to enhancing the capacity to review complex innovative design. So because of that commitment, the CID pilot program was announced in August 2018. Now, this pilot program, meeting program, is an effort led by the Office of Biostatistics at CDER and OBE at CBER. It aims to facilitate and advance the use of complex adaptive Bayesian and other novel trial designs in medical product development. So a common feature of many of the CIDs is the need for simulations to estimate trial operating characteristics. So why do we need to innovate? We have a need to maximize clinical trial efficiency, especially nowadays, while using scientifically sound methods to determine the optimal design for the question or population of interest. Now, CID has the most potential benefit in areas where traditional clinical trial approaches might not be feasible. So, for example, in the case wherein the population size is small or limited, or where there is an unmet medical need. It might also improve decision-making when reliable prior information is available. So, however, we have to ensure that this type of trials will be able to answer the relevant questions and provide regulators with information needed to make decisions. So, an example of a CID is a master protocol. So what is a master protocol? It is a trial designed with multiple diseases, multiple patient subgroups defined using a biomarker, or it could be multiple therapies studied under one overarching protocol. Now, there are two common types of trial designs that fall under master protocol. It could be basket trials or umbrella trials. Now, I decided to use this as an example of a CID because if most of you, might have heard, an example of it is the 4 adaptive master protocols called Active 1, Active 2, Active 3 to Active 4 that was developed by the National Institute of Health, or NIH-led Accelerating COVID-19 Therapeutic Interventions and Vaccines, or called Active, which is a public-private partnership that was formed to investigate COVID treatments and vaccines. So the areas of innovation with master protocol includes the establishment of a trial network with an infrastructure in place to streamline trial logistics, improve data quality, and facilitate data sharing and new data collection. So in this case, it involves the development of a common protocol for the network that incorporates innovative statistical approaches to study design and data analysis. So again, I'd like to emphasize that careful planning of critical elements of the study design is needed for the successful execution of adaptive and Bayesian designs. This is where statisticians, we can take a lead in setting a strategy on the evaluation and implementation of innovative approaches. So here, as we all know, for this type of designs, extensive simulations are needed prior to protocol approval. It is a fundamental tool to explore and understand the operating characteristics, statistical properties, and adaptive decisions embedded within the different design scenarios that we are evaluating. Also, there is a need to establish procedures to ensure that we minimize operational bias in order to protect trial integrity. So we encourage sponsors to have early discussions with the FDA during development of such trials. So what is the statistician's role in reviewing submissions to the CID pilot program? So here, the statistical reviewer or analyst examines the sponsor's submitted simulation code. They also perform their own independent confirmation of the simulation results and conclusions, and also explore additional scenarios to assess the sensitivity of the assumptions made by the sponsor. So they investigate additional parameter settings, and if it uses Bayesian designs, we also look at different alternative priors. So reviewing CID submissions is very resource intensive for the FDA in terms of workload, timelines, and computing resources. In addition to computing resources, FDA needs to ensure that the reviewers have the necessary skills and training to review the simulations. So far, from all the submissions we've gotten for the CID pilot program, the companies or the sponsors have implemented their submissions using R. So here are some challenges to the CID challenges that we could help investigate as statisticians, and as well as provide leadership. So the first is we have to establish a consistent approach on the use of external data sources. How do we combine these different data sources? How do we choose the priors? We also have to have procedures to evaluate the quality of external data. So also, how do we evaluate trial success under a Bayesian framework? And more importantly, how do we alleviate the risk of compromising the clinical trial integrity when the modification of the trial parameters are based on unblinded data? So those are some of the important things that we have to consider with a CID. So the challenges. Next is what are the initiatives that FDA has on COVID-19? The current COVID-19 pandemic is having a serious impact, in our lives, as well as the lives of millions of other people around the world. And it was declared as a public health emergency in January 2020, and a national emergency response was issued in March 2020. So here is a graphic of the different initiatives that FDA has to respond to COVID-19. So one thing I'd like to highlight is in the therapeutics, which is FDA created a special emergency program for possible therapies named Coronavirus Treatment Acceleration Program, or CTAP. It serves as the administrative backbone to ensure that promising treatments are directed to the review divisions in a timely and efficient manner, and to move new treatments to patients as quickly as possible, while at the same time looking whether it's safe or harmful. Now, FDA staff has been working round the clock to review promising applications in record time and continue to optimize the review process whenever possible. So here are some of the COVID-19 guidances issued by the FDA. Specifically, in March 2020, recognizing that COVID-19 would impact the conduct of trials, not necessarily for testing COVID-19, but just trials conducted during COVID-19, FDA issued this trial guidance, and it provides, general considerations on what to assist the sponsors on how to assure patient safety, how do you maintain compliance, and how do you still practice good clinical practice, or GCP, while you minimize the risk to the integrity of the trial. So, as I've said, COVID-19 is a global pandemic that had significant impact on how we conduct clinical trials. It has accelerated the trend towards the use of real-world evidence, innovative trial designs, digital health technologies, and decentralized clinical trials. We can expect that this type of trials will be utilized more in the future, and given the rapid advances in technology, I believe the clinical trials conducted in the next 5 years might incorporate more advanced and easy-to-use technologies to obtain subject data. So this would lead to new data sources, which would provide statisticians and data scientists with new areas of research and will result in the development of new statistical methods or data visualization tools to view and evaluate these new types of data. So here in this slide is some of the COVID-19 initiatives that my AIS team has worked on. So we helped develop a dashboard on the critical care drugs data, which is features a multifaceted signal detection system with elements of machine learning, statistical modeling, and advanced analytics to explore drug sales or prescription dispensing trends. We also worked on an R-Shiny app that looks at the futility criteria for early safety interim analysis. And also we worked on an R-Shiny app that will compute expected number of events along with measures of uncertainty around the expected count for binomial distributions. Not on the slide, but currently we're also looking at different projects using R or R-Shiny that is related to hand sanitizers, as well as using NLP to prioritize field alert reports that the FDA receives for either drug products or label misuse or product contamination. So to summarize, statisticians and data scientists will play a more prominent role in drug development in view of new innovations and evolving regulatory landscapes. We as statisticians need to be effective leaders and communicators in disciplinary teams. COVID-19 has accelerated the use and understanding of real-world data and real-world evidence, novel DHTs and CIDs. And lastly, with the need to manage and visualize and analyze data from various data sources, the development and use of R-based computational tools and applications will see tremendous growth. So, and here's my acknowledgement slides. Thank you for this opportunity.
Great, thanks a lot, Matilde. I'm sorry I didn't get a chance to introduce you earlier.
No worries.
Please. Yeah, so there were a number of questions that came through in the chat, and we'll just have time for a couple of these maybe. So the first one is from Raul Torres, who asks, unlike medicines, algorithms can be easily replicated and modified. What safeguards are in place to prevent this from happening at the FDA? Do you imagine the FDA will relax their strict requirements for full disclosure of proprietary algorithms for DHTs and SAMD applications?
I think that question is referring to DHTs to be used as in the medical devices. So I'm more in CEDAR rather than CDRH. So I don't know if I could give a good answer to that question. I'm sorry. Okay. But I mean, there are guidances that have been issued on those things.
All right, the next question is from Mike Smith, who asks, for CID studies, how do you ensure appropriate statistical and programmatic review for simulation and operating characteristic assessments? There are often complex and intertwined issues.
So for those types of submissions of CID, there is a main group established in the Office of Biostatistics and also with collaboration with the clinical counterparts that looks at all the submissions and in the pilot program. So there's a common group committee that looks into the submissions, and then each of these submissions that are accepted to the program, we have statisticians assigned to look into the submissions. So I believe, like I said, there are several things we look at in submit. We try to independently verify the submissions and also the simulation submissions, and also prior to the submissions of a study, there are 2 meetings allowed for a CID submission. The first meeting is in general discussions with the FDA on what are the things we would like to see in the submissions and what are the different scenarios that would be appropriate to look into for a given adaptive design. So I believe that there are a lot of interactions that is available for sponsors to meet with the FDA before they actually do their studies, to ensure that the results of their studies will be acceptable to the FDA, or thus, the event would be able to provide us with the information we need for our submission.
Great, thanks. So one more quick question, and the rest of these questions I will forward to Matilde, and then she can follow up offline during the break. So we'll go with Paulo Bargo's question. What is the current status of guidelines definition for DHT?
So it's asking for the status of a guidance. I don't think I can either comment. I think I'm not allowed to say if there's something in development or not in development, or will be coming out or not coming out. That's the first thing I learned when I joined the FDA, that I can't say anything about any guidances. So I don't know.
All right, thanks.
Sorry about that.
Yeah, I'll forward the rest of these to you offline.
Okay.
Thanks so much.
Thank you.